We are working, or we would like to know about how microbes work, and how they communicate within microbiome. And to do it, we basically split it into 4 work packages coming from the most simplistic one, looking to the words and sentences. You can use this idiom to imagine the biologically active molecules, chemical molecules, metabolites, but as well proteins. And this is what we are doing in work package 1. So we are taking here a very simplistic model, accompanied by cutting-edge technologies, which we have here, which our partners at university have, and we combine this with genetic interventions into these microbes. And you know, for work package 1, the most important thing is this cutting-edge technologies, this toolkit. And we are happy in the Institute of Microbiology to have the cutting-edge mass spectrometry already. And it's a group assembled around Professor Vladimír Havlíček and currently as well Dominika Luptáková, which basically started to build here the imaging mass spectrometry team. And we are happy to have them in their project and basically implement their knowledge for really finding out these signals, these chemical signals. Nevertheless, we are as well starting something novel here. We want to this project to be something, and you start new looking into the microbe communication. And we have here Jana Kamanová, and he is actually in charge of the metabolomic team within this project. And all of you, I believe, will be collaborating with him. And he basically is within this project installing a new LCMS machine, and we are really looking forward to it. And then, of course, it's not only mass spectrometry, but we want as well to look at the microbes. I always believe that the most important thing, or the seeing is believing. It's a very cliche, but I believe it's true. And for this, we as well planned here an investment cost for a new confocal microscopy, which will be installed here in the institute. So it's and in charge of this instrument, it's basically Ivana and Andrzej from our lab, and we are closely collaborating, or we are closely discussing with Honza Svoboda, and we are in a process of really trying to build here a new microscope facility, or arrange microscope facility within institute, which would hopefully enable much more microscopy being introduced into this institute. And I wanted to, if I can ask for the next slide, and it's incredible, right? And I of course I don't have time to go to all of the details, but like I believe the work package 1 benefits from our expertise, from our models that we have been working many years, and and I will just now go very briefly. I took this opportunity maybe to post the photos. You probably know some of the heads or the leaders, but not everyone. And I really took the people which are employed on this, and I tried to get their photos, but of course I'm missing as well some. So within like the first objective, as I mentioned, we are looking the signal molecules and cascades in bacterial cell-to-cell signaling, and basically it's a project originating in my lab, and we are looking on Aeromonas, and we benefit here that Aeromonas is a symbiont of fish, and we have a collaboration with Dr. Bartůněk, and we are basically able to introduce it into zebrafish and observe it in situ. The other sub-project, it is actually the bacteria that lab of Pavel Braný have been working, and they are looking on the adaptation of this Staphylococcus aureus and Streptococcus pneumoniae, and studying how it adapts to host cells, but as well to the environment. What are dormant stages? What are the clues that awake this bacteria? And if I may ask, and then we we as well have here, we are trying to discover something novel. And we have here a research built around previous work of Gabina Novotná, who actually starting to realize that antibiotics are not only for killing bacteria, but within microbiomes, they might play as well a signaling role. And it's a theme around her, which we have in this project. And as well, we are happy to have Ondřej Černý, who basically spent his postdoctoral stay at Imperial College London, and he introduced here a topic, heterogeneity. So if you remember, like microbiome, it's not a clone of bacteria. It's separate bacteria, separate entities, and they divide the work. And Ondřej is actually looking at the division of work in bacterial societies and how they communicate and what are the metabolites regulating it. And if I may ask, and you know, sometimes we work on the small molecules, but if we think about signals, we as well have to think about proteins. And within this institute, we have a very, very good research on the structure-function relationship. And we didn't want to miss it in our project, and that's why there is a separate objective focusing on proteins. And it's actually a research built around Professor Šebo, but currently as well taken over by Láďa Bumba and Jiří Maší, and as well Radim Osička, who is not part in this project. And we are really, and as well I came from lab of Peter Šebo, so it's my expertise, and we are here looking on the substrates of the effectors of types of the secretion system, type 3, type 6, type 1. And if I may ask, and then of course I mentioned small metabolites, I mentioned proteins, but then we as well within microbial societies we think about nutrition, and it's objective built around Andrea Palizová and Dominika Luptáková, and she is looking how microbe actually use ionophores. For example, it can happen that a certain microbe produces ionophore and another bacteria can use it. And if I may ask, and we have 2 more slides and that will be it, but I don't know whether you will be stuck here forever for the next leaders. So and then we are as well happy to have within this project the former group of Professor Vladimír Křen, currently headed by Katka Valentová. And within this group, we do have very nice objectives or research objective leader. It's Pavla Bojarová, basically focusing on the sugars, on the human milk oligosaccharides and their effects on the gut bacteria, and as well Kristýna Slámová focusing on the oligomers and their modifications. And then of course we have Katka Valentová and Professor Křen, and they built for many years. They have building in their labs enzymes to produce novel bioactive molecules. And within this project, they want to screen them as well in cooperation with VŠHT, with Jitka Viktorová, who has this robot for screening them, and as well with the help of Professor Štěpánek, that we will build, trying to pack them into some new delivery systems and try to make a difference. And I will give with this, I will give floor to František Štěpánek, and he will tell us about what we can do with these molecules when we find them and how can they be delivered.